Started. It's time. So we will go ahead with the series of talks here. And each speaker has 10 minutes, as you know, and we can have questions right after each talk. So we will have time for a couple of questions after each of the speakers. So the first speaker is Dr. Zibing Jin, who will be speaking on generation of vitreoretinal organoids from pluripotent stem cells. Is Dr. Jin here?
Dr. Jin is not here. I think he has a pre-recorded talk.
He has a pre-recorded talk.
Could we play his pre-recorded talk? Dr. Jin is not here with us. Could we play his pre-recorded talk? Does he have one?
Is who has a talk? It's not ready. Okay. So that's not possible, right? So Dr. Jin is not here. Yeah, he's not at the meeting, so we will move on to the second talk in the list, which is Dr. Swaminathan, who will present on behalf of Dr. Arko Ghosh on novel AAV vector designs retinal dystrophy.
Thank you. It's coming up. Yeah. Good morning, everyone. And sorry, I'm here on behalf of Arko Ghosh. He's the brainchild behind our gene therapy program. He sends his apologies. He's very unwell, or else he would have been here. And I'm part of his team, but I'm trying to fill his very big shoes. So pardon me if I just falter. Just to start, we are from a city called Bangalore in the state of Karnataka. Apart from good science, the state also has some beautiful coastlines and mountain ranges if you wish to visit when you're free. So this is going to be the brief outline of how this talk is going to be split. So the introduction is going to be on gene therapy. The main aim of gene therapy is to provide patients with a healthy version of the gene using the currently available 4 different strategies. You can replace by gene replacement or augmentation using the functional copy of the gene, or you can perform gene editing of the mutated gene, or gene silencing using degrading the miRNA and modify the gene therapy sequence by altering the expressions. And some of them are already in clinical trials. This is just the different strategies. The reason why I'm telling you this strategy is in vivo gene therapy, it's mostly vector-based, and viral vectors play a very important role in delivering the functional copy of the gene. And adeno-associated virus today is going to be our focus of discussion. From a very humble beginning as a contaminant, it just evolved itself to be one of the preferred viral vectors in gene therapy, the adeno-associated virus. And there's already approved drugs based on adeno-associated virus for gene therapy applications, quite a few over there, which you can see. The reason why it became so popular is because of its efficient delivery of genes in both dividing and non-dividing cells, and its high level of the transgene expression, and particularly very low immunogenic, and the most important is non-integrating, unlike the lentiviral system. So these are the salient features as to how AAV became one of the choices in gene therapy applications. And the advantage here, what we have is its serotype and tropism. Okay, we have multiple serotypes, and each of these serotypes have specificity towards certain targets and tissues. And we have quite a range for its tropism towards the eye and various tissues in the eye. But the most important part is how do we optimize these vector designs in order to improve this efficiency. One may ask, why is there a need for such a optimization process? One of the hurdles with AAV vectors is its packaging capacity. It's approximately 4.7 kb, and some of the genes may be beyond that. So that is one of the major packaging capacity. And the second one is if you increase their transduction efficiency, you can reduce the amount of the viral vectors you're going to inject. That will bring down however low immunogenic it is, it still may have some immunogenic response. So reducing the dose is always going to be helpful. And if you reduce the dose, you also bring down the cost. So these are the two hurdles which we are trying to address using vector designing system. And the two examples which Arko wished to show you today is using a dual hybrid vector system for transgene engineering to address the packaging capacity, and also to improve the transduction efficiency using the capsid engineering process using insertion peptides. Two examples. And his lab, our lab, but he primarily drives it, has mastered the technique of improving the transgene processes as well as the capsid processes, variety of them. And we have the entire vector production facility in process as well. So the first example is the transgene engineering, that is a dual AAV vector. How do we improve its AAV vector is used for a bigger gene. So many of these genes as listed here may not be suitable because of the AAV's low packaging capacity. To address that, the strategy which has been followed around the world and including in our lab is that using the truncated or the split gene process, where we split a gene into two parts and then let it recombine inside the target cell. There are various strategies. It's called the dual vector strategies. You have overlapping vector, transsplicing vector, hybrid vectors, and NTN containing vectors as well. Today I'm going to show you an example of what we do with hybrid vectors, for which Arko has the patent for. This is a very simplified version of what the dual vector strategy is: to take a piece of genes, split them into two, package them into two different AAV virions, and inject it into your target cell or tissue, and they get reconstituted inside the cell. So how does it actually work? So a single gene is split into two parts, and it gets packaged in two different virions. The first way, it utilizes the recombinogenic capacity at the DNA level to combine these two genes. One is via the homologous recombination, and where you have a spliced donor site and a spliced acceptor sites, and there is an overlapping region. And once they come into the cells, the central part gets spliced out, and you have a mature RNA for your target gene. The other way is where the recombination happens at the ITR level. Okay, so these are the two main strategies in which you have a complete formation of your intended RNA, which then translates into proteins. But in this as well, how the transgene engineering was optimized was putting these two overlapping sequences, or optimized overlapping sequences. This one is highly recombinogenic. This one, in addition to being highly recombinogenic, the splice also has splice enhancing introns, so that it removes these regions during the ITR combination. And some of the examples we wanted to show is using the Stargardt's disease, and we are very familiar with what happens with Stargardt's disease, along with lipofuscin accumulation, drusen formation. You lose your central vision, and also in the Indian cohort, we do have a lot of ABCA4 mutations in the patients. So we are going to use that as an example to show how the dual vector strategy can be very useful. So what you see here is the retinal fundus autofluorescence image of a normal person, and you have the Stargardt's patient, and you can see enhanced autofluorescence. And in the ABCA4 knockout animals, you can see the increased autofluorescence as well compared to the wild type. So using this animal model, and we are using blue light to induce the retinal degeneration, because normally it doesn't degenerate by itself. So the BL stands for blue light induced degeneration. When you use these two, this is your control AAV treatment, where you only have one half of the gene, because you are using the dual hybrid vector. So in these two, one is the splice enhanced introns and the overlapping alkaline phosphatase sequence. You can see a very good reduction in the autofluorescence compared to your wild type as well as your control group. this clearly shows the dual vector strategy actually applies and works very well in the system. So I'm not showing you some of the in vitro results, but we're jumping and showing the whole thing in the in vivo setup. And not only does it reduce the autofluorescence, it also regains and maintains the retinal architecture as well. So the animal develops this retinal degeneration following blue light in 4 to 6 months. So all the injections happened during that time period. And it was left for 3 months for us to see the endpoint results. Both structural and after the structural things, we wanted to see the functional aspect of the process, and what you see here is the ERG, the electroretinogram. You can see the controls, that is the ABCA4 knockout ERG plots, and what you can see is that in both the cases of both SEI, that is the splice enhancer introns dual vectors, as well as the overlapping alkaline phosphatase dual vectors, you can see the return of a normal ERG activity in these animals. And this is this shows you the quantification of the ERG. This helps you understand and appreciate that the SEI, which is both SEI and the OAP system are highly recombinogenic. One is far more superior because of the splice enhancement introns. You can see the improvement is much better than the other one. And in addition to that, what was observed was after the vector transfection in these animals, you can see increased ABCA4 gene. Along with it, you can see increase in level of opsins as well. Okay, and the SEI group showed a bit slightly higher opsin levels compared to the OAP groups. So that is the first example of the transgene engineering. And the next one we would want to give an example of capsid engineering is to improve the transduction efficiency. Here what we have done is we used a rational design approach, and we have taken, so that's 8 minutes? Oh, I see. Okay, so should I?
Time is.
I'll take 1 minute.
Yeah.
It's almost done. So we have an insertion peptide into the capsid, and we have 2 of them. Don't worry about the C and T. This is our internal naming. Insertional peptides were included into the capsid. The capsid integrity was good, and all the quality controls was checked. And what you can see here in AAV2 in the animals after 6 weeks, you can see an increase in the GFP expression when you have the insertional peptides compared to the wild type AAV2. A similar example is for AAV9. When we inserted a peptide in the capsid, it enhanced the ability to transduce, and you can see the expression has gone up. And with that, the summary is we do have a good dual vector system as well as capsid engineering system to improve transduction efficiency. And the next couple of slides is just pictures to show where we are based. That's the hospital, and that's the center which ARCO leads. And that is the Class 100 facility, which has been recently approved for clinical grade vector production. And he has the entire process in place. And these are the clinical trials, which one of them is approved and the other ones are in preparation. Along with it, other preclinical studies are ongoing, just not for the eye disease, but it's beyond as well. Both sickle cell as well as muscular dystrophy is being handled. We appreciate the entire team funding, late founding chairman, and our current chairman and vice chairman, along with the entire Grow Lab, the gene therapy team, who we acknowledge for their tireless work, and all the funding support which supports ARCO in this mission. Thanks very much. Sorry, I exceeded the time.
One quick question,
Yes,
if
please.
anybody has.
Oh.
Yeah.
Thank you. Wonderful.
Thank you.
I'm Hemant Khanna, Astellas Pharma. I wanted to ask you about a couple of things. One was I'm assuming you did subretinal injections, correct?
That's correct, yes.
Could you give us some idea on when you did the ERGs after you injected your...
3 months. 3
no,
months
but I mean, the amplitude, did it reach the wild type levels, or was it... Because I'm assuming it's regular ERG, or is it...
No, it's animal.
Yeah, it's full field ERG,
Yes,
correct?
yes.
So did it reach wild type levels, or was it less than that?
I don't know that.
Okay.
Yeah.
And in this, could you also...
ARCO would have that data, yes.
That's okay. I think I was just remembering what RP65 had
I see.
in mice, and there were some issues because you did one subretinal injection, right?
Yes, one.
Yeah, so I don't know if that may reach the wild type levels. Just wanted to get your opinion on that.
Right.
The other one was, I like that idea of having those two split parts, but what do you recommend for genes which are even bigger that even that strategy may not work? Like you had written USH2A, EYS. I'm not sure about EYS, but I definitely know USH2A and Duchenne. Those may not be feasible even to split them.
Right. I'll tell you what ARCO has in mind. I don't have full authority on that. He does these concept called mini genes. He takes the most functional part and tries to do that rather than the entire protein.
I got it. Thank you.
Yeah, thanks.
Okay, you have a question? Yeah.
So do you mind if I ask a question? So in your data, you compared SEI and OAP in terms of functional rescue with full-field ERG,
Yes.
and SEI was better than OAP. Just hoping to get your insights as to why you think that might be. Why would one approach be better than the other in terms of functional rescue?
So the splice enhancer introns helps in increasing the efficiency of stabilizing the mRNA as well. So he said it's intron-mediated enhancement. So there are more transgene there than the OAP. There are other data which supports that.
I see. Thank you.
Thanks.
Okay, so I think we can move on to the next talk, and that is by Dr. Sue on retinal transplant immunology.
Okay, good morning, everyone. Thank you to the organizers for inviting me to come to give this talk. So I'm from Singapore, and I'm Xin Yi. So these are my financial disclosures. So in my lab, essentially, we've been looking at different strategies to treat end-stage retinal degenerative diseases. So one approach is to develop tools for surgical transplantation. Second is to understand whether we can augment the survival of these RPE cells post-transplant. And here today, I'm going to talk a little bit about a novel hyper-autoimmune stem cell resource that we've been working on. And more recently, we've also gone a little bit into gene and RNA therapy. So the disease that we're interested in today is age-related macular degeneration. I think most of us are familiar that it is one of the leading causes of blindness globally, particularly in the elderly. And fortunately for us as ophthalmologists, there are multiple treatment opportunities for AMD, particularly for dry AMD. Now we've got the complementary inhibitors. For wet AMD, we have anti-VEGFs. But however, as clinicians, often many of the patients do come to us with end-stage disease, like geographic atrophy and a disciform scar, for which we currently do not have effective treatments. And this is where I think cell therapy would be a very good approach. And particularly for AMD, what we're interested in is the retinal pigment epithelial cells over there. And the RPE cells are crucial for AMD because they support the function of the photoreceptors. So some of these are as a schematic diagram showing the roles of RPE cells. Importantly, they help to turn over the photoreceptor outer segments once every 10 days. As you can imagine, they are working really hard, and because they're terminally differentiated and non-renewable, they start to accumulate DNA damage and oxidative damage over time. And as they begin to kind of be on a downward trend and they start to have dysfunction, that drives AMD. So the idea for stem cell therapy for macular degeneration is a very simple one. You get stem cells, you differentiate them into RPE cells, and then you transplant them into the eye. So here what we're thinking of now is to replace RPE cells that are non-functional with healthy RPE cells and hopefully be able to recover some of the vision. So this is not a new idea. There are multiple high-profile publications that have been out there in the US and the UK, and particularly they are looking at using autologous cells. So here meaning they take cells from the patients, they reprogram them into RPE cells, and then they inject back to the same patients. This will help to overcome immune rejection. Alternatively, many of the groups have also looked at using human embryonic cells. And I think more encouraging is that more recently there is a clinical trial by Lineage Cell Therapeutics indeed saying and showing that transplantation of embryonic-derived RPE cells can indeed improve vision as well as regain some of the anatomical structures. However, there are still remaining challenges to bring this into clinical reality. I think one of which I alluded to earlier is the optimal surgical approach. Do we do cell suspension? Do we do monolayer transplant? I think very importantly also is to develop an ideal cell resource. So for embryonic derived stem cells, it's a limited resource and potentially ethical concerns because we're deriving it from an embryo. For induced pluripotent stem cells, there are two large categories. One is autologous, which I alluded to earlier, where you take cells from your own patient, reprogram them, and inject it back. And you can imagine this is a very laborious process where you need to develop one particular cell line for each patient. And these cell lines would have the inherent AMD risk alleles that arising from these patients. The other approach is to use allogenic. Basically, you have a master cell bank, and then you use that to treat patients in a broad manner. However, with that, you have the potential problem of immunogenicity. So you need to do some form of immune suppression as you would do for any sort of organ transplant. And also, more importantly, people are also thinking about we can get the stem cells in, but are they functioning? Do they make the connections? How can we enhance survival and integration of these post-transplanted RPE cells? And this is the topic of our discussion today. So in our lab, we've been working very closely with a biotech company called Cell Research Corp in Singapore. Here, our hypothesis is that if we were to derive stem cells from the cord lining cells, and because pregnancy is an immune miracle, this maternal-fetal interface is immune privileged, and if we get cells from this epithelial cells of the umbilical cord lining cells, perhaps we don't have to use immune suppression as much. And the further advantage of clips is that it's unlimited, so umbilical cords are being thrown away every day, so it's a resource that we can always come to. They are very young cells, they age 0, genetically, somatically pristine, and they have HLA, so we can HLA match them as well. So the idea is that this potential cell source could be a universal cell source that we could use, overcoming the need to use autologous approaches. So the first experiments we did was in vitro. So we have the cord lining cells, CLECs, we reprogram them into IPS, so we call them clips, and then we differentiate them into RPE cells. And you can see on the top here, the clip cells, sorry, it doesn't project very well, sorry. So basically in the parental lines, they have lower expression of MHC class 1 and MHC class 2, and after we differentiate them into RPE cells, and we stimulate them with interferon gamma, we also see that there is reduction in expression of MHC class 1 and 2 compared to the skin-derived and ES-derived cells. And next thing we did was then mix them together in a lymphocyte co-culture. So we mix RPE cells with T cells. Consistently, you can also see that the RPE cells have, they don't stimulate proliferation of both the CD4 cells as well as the CD8 cells. So now we moved on to an in vivo system. Here we are using mice that are humanized, meaning mice with a human immune system. We injected the cells of RPE-derived, ES cells, clips as well as skin-derived, and we look at both a systemic immune response as well as a localized response. So here, in the subcutaneous space, when we explanted the transplants, you can see that the pigmented RPE cells still remain. And then we also look at the serum and measured pro-inflammatory cytokines. Here again, you can see that in purple, for the clips-derived RPE cells, there was a reduction, there was less induction of pro-inflammatory cytokines as well as reduction in T cell activation in the various organs. And we then looked at the localized areas. Here we are looking at human CD45 cells stained in red. So this is a pan-immune cell marker. You can observe here that in the clips on the extreme left-hand side that there was very little infiltration of CD45 cells compared to the ES-derived and the skin-derived RPE transplants. Then we leverage on single-cell technology to kind of understand what are some of the processes that are happening at the cellular level. What we did here was explant the cells and did single-cell transcriptomics. In purple, you can see again, comparing the pre-transplanted to the post-transplanted RPE cells, there was a reduction in upregulation of the MHC class 1 and class 2 compared to the skin-derived cells. And next, we wanted to again understand or characterize the immune cell infiltration in the microenvironment post-transplantation. And here you can see in purple, again for clips, very interestingly, there was a huge population of B cells and myeloid cells compared to the skin derived in the brown and the green, and there was a much less population of T cells. So this again is a very unique immune microenvironment that we observe specifically for the clip cells. Then we dug a little bit deeper to say, what is the population of the T cells that are available there? And you can see in the circle over there that very interestingly for CLIPS, there was a predominance of Tregs. So Tregs is a subpopulation of T cells that have a kind of a hypoimmune property. They regulate other T cells by secreting anti-inflammatory cytokines and then down-regulates the immune response. So again, this was very unique to just the CLIPS RPE cells. And now we are undergoing further analysis to identify immune modulatory factors. And our hypothesis is that the CLIPS RPE cells somehow secrete factors to upregulate the surrounding Treg cells and therefore suppress the immune response in the microenvironment. That was in the subcutaneous space of mice. So we wanted to ask the question, what happens in the subretinal space? So here we use a larger animal in rabbits. We transplanted the monolayers of the CLIPS cells into the rabbit eyes, and we did some phenotypic analysis over there, essentially showing that consistently in the CLIPS RPE cells, the transplants were quite well tolerated. Similarly, we also did single-cell RNA-seq, so we explanted them, compared the in vivo cells to the in vitro cells. And again, the CLIP cells do behave very differently. Again, in purple over there, we saw that they have a higher expression of mature RP markers. So our hypothesis is that because it's transplanted, there's less of an immune response. These RPE cells can then flourish and act as RPE cells. So we looked at gene expression in terms of visual cycle proteins, extracellular structure organization, melanin synthesis, and phagocytosis. Again, you can see in the red arrows there, transplanted CLIP RPE cells are in a way like super RPE cells that can outperform the other RPE cells. And consistently, we saw there was a downregulation of MHC class 1 and 2, which is what we observed already in the in vitro cells, as well as the humanized mouse models. And interestingly, we saw an upregulation of TGF-beta pathway, which we postulate could be one of the mechanisms in which these cells are different from others. So last of all, we went to a non-human primate model. So here we transplanted CLIPS RPE cells, compared the skin RPE cells into non-human primate in the macular area without without immunosuppression. So this is a xenograph. And at 3 months, you can see in the yellow arrows, the CLIPS RPE cells were better tolerated compared to the skin RPE cells. And we have followed this up for up to a year in some of our models, and also have done functional studies, which I will not show here. So in summary, CLIPS RPE cells is potentially a universal source of hypoimmunogenic cells. We've shown that in vitro. We've also showed you some allograft data in a humanized mouse model. We looked at localized immune response, systemic immune response. We have some understanding of the mechanisms, but we're still trying to uncover that, and also shown you some evidence of transplantation into large animal models. And we have filed a provisional patent application for this, and hopefully in the near future, we'll be able to bring it into clinical trials. So I'd like to acknowledge my team, my funding body, and I'll be happy to take any questions. Thank you.
Questions?
Very nice talk. I often have patients tell me that, oh, we saved cord blood after our child's birth. And they say, oh, is there something we can do with it? And I say, I don't really know. What they save, could that be used to make these CLIP cells, or are they just saving, blood cells, or do you get epithelial cells?
Yeah, I think it very much depends on the package that you get. So I've stored my own cells as well, and with a Singapore company, and they give you the option of just saving the cord blood itself or the epithelial cells. So it's very much dependent on your service provider. But what we've also observed is not the same across all cord lining cells. We do see some super performers. And of course, what we've shown you here are the high performers. There is a huge variation across different cord lining cells that we deal with. So I would think that, it wouldn't, we couldn't use the same patient's cord lining cells and assume that they will function as well.
Yeah. Questions? Anybody? Okay, I just have, one query here, that you have actually now manipulated the immune system, and so to an extent the immune response has been, taken
Dampened,
care of.
yeah.
But what about, we're talking about transplanting these cell sheets into dry AMD patients, and there's a milieu of cell death and degeneration there. So if you put these cells into that environment, what would be, it's not an immune-related issue, but generally, so what would be I don't know whether you have any such experience, how long you expect the cells to survive, because, you have that sort of in the recipient.
Yes, yeah.
Yeah.
Thank you very much for your question. So this is exactly one of the things we would like to look into. So we have developed disease models in monkeys. So we're using micropulse to induce localized RPE dysfunction, and that will partially disrupt the Brooks membrane also. So I think we will hope to kind of challenge it a little bit more to see if the transplants in a, in a more immune complex situation, do we still get that same effect? But this is certainly something that we can look into.
Okay, any other questions? Okay, yeah.
Were there HLA mismatched samples?
Yes, they were HLA mismatched because we wanted to push the system a little bit more. Yeah. Great, thank you.
Okay, so we will move on to the next talk, which is lessons learned from gene therapy by Dr. Berokal.
Thank you very much. Actually, my sister is the expert in this topic, but I will talk to you about the vascular polymer experience. So we know that vortigine is Luxturna. It was FDA approved in 2017 for the treatment. Okay. For the treatment of RPG65 mutation associated retinal degeneration. And it is associated with severe visual impairment at birth that can progress to complete visual loss. So, what have we learned about the surgical technique? Well, it is very important to remove the hyaloid, and it tends to be easier if you start near the fovea, as it is shown there. And the flex loop is very useful in inducing the PVD, and you will always be helped if you use triamcinolone. Basically, you can see here how this is done. The other thing that is important is to bevel the tip of the cannula and check for the injection pressure. So here, we're cutting the tip at an angle, and that will make it easier when it's actually penetrating the retina of these patients. So removing the hyaloid and then going in at an angle. Using a microdose injector will give you more controlled delivery than actually using manual. And it is better to make 2 blebs than a very high, thin bleb. And at the beginning, we were detaching the area of the fovea, but now we know that that's not a good idea because you can induce a macular hole. So basically, here you can see doing multiple blebs is actually better than just doing one big bleb. Intraoperative OCT is very useful because you can check the height of the bleb. This is a case where, the fovea was detached and you can see how thin it gets, so you can see how easy it is to actually pop that. 3D surgery is ideal for this type of surgery, the Ingenuity. And here you can see, because you can change some of the parameters to tissue detail mode, which will let you see better. You can use green if you're staining the ILM, and you can see how well, you can see all the structures there. And the staining with the triamcinolone. Detaching the hyaloid in the posterior pole. Checking the periphery, very, very important. You don't want to create any breaks. Here we're beveling the tip, shown before, and then starting the bleb. Some people start the bleb instead of touching the retina by just injecting pressure and sort of penetrating it that way through pressure. So creating multiple blebs is really what you want to do. And at the end, there's always vector that goes into the vitreous cavity. So it is of essence to go on, into air that will help coalesce some of these blebs together, but also removes the vector from the vitreous cavity, which is another very important part of doing that. And if you do the injection instead of manually, where it's very inexact, if you do the injection with the injector, you can control how quickly the fluid is going in. And this is just checking with the intraoperative OCT for that. Going into air, make sure you remove all of the vector. And then subtenon's triamcinolone, besides antibiotics, is very useful in these cases. Using these lenses, it makes it much easier. The Oculus Lens C gives you a much better view when you're doing these surgeries, and they also have a lens that is caught in both edges. So if you're doing a small child, or if you're doing, a person with a prominent nose or whatever, it will allow you to actually get a much better view. There, you can see the view with that lens. It's much better than any of the other lenses, out there. And we have a patient here who has significant, pigmentary disruption, but you can see the periphery really well as you're doing this whole maneuver. Going to air, as I mentioned, to remove all the viral vector is of essence. We want to make sure there's no viral vectors left there. And starting oral steroids 3 days before surgery and subtenous Kenalog at the end of surgery, and intravenous solomedrol at the end of the surgery too. So post-treatments, what are some of the lessons that have been learned? This article was focused on characteristics and growth rates of Luxturna chorioretinal atrophy, which we actually see in some of the areas of injection over a mean of 2 years. And 14 patients were followed for 27 years. And we see several, types of atrophy. The touchdown, which is at the injection site, the nummular, as you can see, and B, perifoveal, and mixed, which is a combination of all of the above. Growth rates for each atrophy subtype outpaced multifocal geographic atrophy in AMD. Perifoveal is the fastest growth rate, nummular intermediate, and the touchdown the slowest. At Bascom Palmer, they have a total of 13 pediatric patients. 26 eyes were followed for 5 years. 9 of the 26, 34%, went on to develop chorioretinal atrophy. The patient characteristics were a mean age of 8. Atrophy-positive patients, the mean was 9.4 years of age. So basically, the hypothesis for this is that tissue indentation from the injection cannula causes that, shear forces from the fluid propagation, the speed and the force that we're doing this, trauma from the transient retinal detachment, vector toxicity, target overproduction, inflammation, and other mutations. So we see chorioretinal atrophy in 35% of treated eyes in children. Touch atrophy was the most common type. Nummular lesions were typically the largest lesion type, perifoveal, and nummular atrophy most likely to progress. Risk factors were most patients were all myopic, with an average of -4 and -3 in the left eye. So the implications gave us some surgical technique modifications, avoid foveal detachments because we don't want atrophy in the fovea and we don't want to create macular holes, minimize the RPE touch, so sometimes we're using the fluid to penetrate into the retina, use automated injectors, decrease the speed, suppress surgical and drug inflammation, maintain oral steroids pre and post-surgery, posterior subtenons, intraoperative solomedrol, and topical prednisone and difluprednate. So these are the cases. You can see here. So this is actually the first eye of the surgery, and you can see this kid. I think it's playing. It's not playing. Anyway, and this is another little girl. This is the US, so they play with guns. So you can see how, she could actually aim at the target pretty well. So thank you very much. And this is my sister's team at Bascom Palmer.
Open for questions. Yep.
Maria, thanks for a great talk. So for the pigmentation after the vector delivery, is it associated with poor vision in the patient? And what does that imply? Does that imply actually it's a good sign that RPE cell were activated or functioning after the transduction of the viral vector, or is a toxic effect? What's the current understanding about this?
Well, that's what is not really known. They think it's more of a damage. We do see this pigmentation in many of the other gene therapies that occur. If it's at the site, we know that it's actually trauma from the injection site. But that's why the vector is not being injected in the area of the fovea. Okay, that's why the modifications of the technique occurred.
Anyone else has a question? Well, I have a comment. I mean, it's kind of interesting and very revealing to me to see what you presented. One of the things that we read, now I'm not a clinician, so I'm not the expert on this aspect, but one of the things we read in terms of the RPE65 gene therapy that was carried out was the best visual effect was, in fact, in the area of the injection, just surrounding the site of injection. And in fact, it didn't have a really large spread beyond that immediate vicinity of the injection site. So is this going to sort of cancel that, whatever benefit might be obtained in terms of the gene therapy vector, Because, see, what you show is that damage occurs right there and it's related to...
but it doesn't occur in all patients.
Yeah.
You see, which is what's interesting. So they don't know if it's the force that the retina is being detached, shear forces. So there's a lot we don't really know about gene therapy, really.
Okay.
As a follow-up question, because you mentioned that you would avoid the foveal area, do you still see improvement in function over the foveal area, even though the injection sites are peripheral?
Yes, because it's injected in the posterior pole, but not the the problem with the foveal area, and you have it any time that you inject subretinally, is that if there's a lot of fluid, you can create a macular hole in those patients, which would compound the problem.
Thank you.
Thank you.
Thanks, Maria. So we'd like to invite the next speaker, Dr. Xiaoying Tan from Hong Kong, to give the next talk. You have the pre-recorded talk? Yeah, okay. So she's unable to come, so we'll have the pre-recorded talk. Thanks.
Hello, I'm Xiaoying from Hong Kong PolyU. I would like to extend my gratitude to the organizing committee for including me. Unfortunately, I'm unable to attend this meeting in person. I want to take this opportunity to send my greetings to all of you and wish the conference a great success. And here I will present the topic on cellular and molecular approaches to treatment of neuromyelitis optica spectrum disorders. The projects are supported by several research grants, and there's no financial interest in this presentation. Neuromyelitis optica spectrum disorders, NMOSD, is a severe central nerve system autoimmune inflammatory disease that can cause the demyelination lesions and neuron injury. Myelitis and optic neuritis are two of the main manifestations. The current treatment options are limited. Poor visual outcomes and a high number of recurrent attacks are found in NMOSD patients, even after long-term therapy. The prevalence of NMOSD is from 0.5 to 10 cases per 100,000 people, but it is more common in Asia population. In terms of the mechanism, it is widely accepted that ACOP4 IgG, which is a specific antibody produced by the serum B cells and plasma cells, penetrate into the central nerve system, resulting in binding in the ACOP4 channels on astrocytes, and then lead to the complement-dependent cytotoxicity, eventually damage to the astrocytes, oligodendrocytes, leading to demyelination and neural loss. The current clinical management, including the treatment in the acute phase to alleviate the symptoms and the remission phase to prevent the relapse. The patients are usually treated with high-dose steroid and immunosuppressive therapy targeting the production of the antibodies. However, the efficacy is limited. More than half of the patients remains poor vision or suffering recurrence. The long-term systemic therapy causes several side effects and complications, also influence patients' quality of life. So it is necessary to develop new and effective approaches to treat NMOSD. This diagram shows again the mechanism on the ACOP4 antibody produced and passed through the ABB and then combined with the ACOP4 on the astrocytes, so that the targets of the medications are going along with the pathway. For example, the current therapeutic drugs are mainly targeting to the B cells and plasma cells. The development of more new agents is ongoing. Some of them have entered the phase 2 and 3 clinical trials. Our team has conducted serious clinical studies on the efficacy of those treatment options and new medications, such as the rituximab. And in this presentation, I will focus on the work of cellular and molecular approaches and introduce the one we have attempted blocking the binding of ACOP4 antibody to the ACOP4 channels. Well, the protein structure of the ACOP4 consists of 3 extracellular loops, loop A, C, and E. Therefore, the combination between the antibody and the ACOP4 protein must be on these 3 loops. The peptide sequencing of the 3 loops are known. We have the idea that design and synthesize the 3 loops in vitro and use these recombinant peptides to neutralize the ACOP4 antibody in serum. Based on this idea, we conducted several experiments. First, it has been proven that ACOP4 IgG concentration detected by ELISA in the samples of the patient's serum can be significantly reduced by the pre-mixture with the mannitol peptides, especially with the loop A and loop C, and the mixture of the loop C, ACE. This data demonstrates that ACOP4 IgG in patient serum can be recognized and neutralized by the mannitol peptides. We performed the cytotoxicity IC and live cell immunofluorescence experiments in the primary astrocytes culture, which also suggests that the mannitol peptides could reduce the cytotoxicity of the ACOP4 IgG to the astrocytes. We further tested whether the mannitol peptide could block the binding on spinal cord tissue from Louis rats. The results consistently show that the loop A and C significantly reduce the human IgG labeled area at the spinal cord. Our data implied that the mannitol peptides were able to reduce the cytotoxicity of patient serum to the mainline of the spinal cord. We then tested and confirmed the protective effects of the mannitol peptides in our in vivo rat model by the functional and the structural assessments. We can see the graph showing that the loop C significantly reduced the binding of ACOP4 IgG to the spinal cord tissue and inhibit motor function impairments. However, the current understanding of NMOSD pathogenesis comes largely from research conducted in the brain and spinal cord, but not the optic nerves. Questions have been raised about whether the pathogenic mechanism in the brain and spinal cord also applies to the NMOSD-ON. Spinal nerves and optic nerves are essentially different. The spinal cord contains both motor and sensory nerves, but optic nerves are only sensory nerves. The particularly high sensitivity of the optic nerves to the ACOP4 IgG in AIOS2025-GP32 is still unexplained, and it is not yet known whether the pathogenic epitope of ACOP4 are also localized at the extracellular loop C on the astrocytes around the optic nerves. The effects of synthesized ACOP4 mannitop peptides in reduction of the neurocytotoxicity in MOSD-ON are also need to be demonstrated. So that we continue our studies specifically to the optic nerves, so-called NMOSD-ON. Firstly, we tested whether the mannitop peptide could block the binding of ACOP4 IgG to extraviral optic nerve from the rat. The results shows that all the mannitop significantly reduced the human IgG labeled area. We also tested in the mouse optic nerve. Results showed that the M loop C and E significantly reduced the human IgG labeled area, implied that the ACOP4 mannitop peptides were able to reduce the cytotoxicity of the ACOP4 IgG to the optic nerve, which is slightly different from the result from the rats. Further studies are needed to address the reasons. So far, we have tested the effect of synthesized ACOP4 mannitop peptides in reducing ACOP4 IgG cytotoxicity to ex vivo optic nerves. We have successfully established an MOSD-ON mouse model by intracranial injection of the ACOP4 IgG on the optic nerves. I have another presentation to introduce this model, and for those who are maybe interested, please refer our published paper. So the next step for us is to validate the effect of synthesized ACOP4 mannitop peptides in our in vivo mouse model. Hope I can share with you the results next time. So I would like to summarize some points at the end of my presentation. MOSD is a severe autoimmune inflammatory disease, and the current treatment options remains limited. Ongoing research is focused on developing new agents. We have developed a novel approach to inhibit the binding of ACOP4 antibody to ACOP4 by synthesizing ACOP4 mannitop peptides. Our preliminary results contribute to understanding the pathogenesis of MOSD, and this targeted therapeutic strategy aim to enhance treatment efficacy and improve patients' outcomes. Finally, I would like to thank all my teammates and colleagues in Hong Kong PolyU and my collaborators from the CHK and mainland China. Thank you very much, and I hope you all have a fruitful time in New Delhi.
Right, we'd like to invite the next speaker, Dr. Hemant Khanna, to share with us on the clinical applications in gene therapy of ocular diseases. I think you can use this over here, I think. Oh.
Thank you very much. I want to thank the organizers for giving me the opportunity to talk about gene therapy. So I'm from Astellas Pharma. We do have some gene therapy programs and also small molecule programs. And what I thought I would do today is we have heard a lot about very great talks on how good the results can be with gene therapy. But there are also programs which do not make it to either to clinic or do not make it even at early stage of discovery or even development. And it's always a good idea after a few years to really go back and see how we started so that we get some encouragement and we keep going. And that is what I thought. I'll start with some background, a little bit of background. You may already know this, may not know this, but I found it very interesting that the idea of gene therapy actually started in the time when Liederberg, Beadle, and Tatum, they were the pioneers, Nobel Prize laureates who showed bacterial genetic recombination, viral transduction, and from this was an article where Liederberg actually said that there could be an application in molecular biology if we could graft polynucleotide sequences onto a virus DNA and then take it further. So that's started from where I think this is where the field started. And then again, there was talk about, there is excitement, but then there was also issues with what are we going to do with humans, because this is genetic manipulation to some extent. But then this field kept going, and then there were a few papers that came out that actually could transfer genes into cultured mouse cells and also in stem cells. So that all was very encouraging. That helped quite a bit to take the field further, but as always happens, there are people who actually take it too far, too quickly, which is in a way good and bad. I mean, nobody nowadays will never do this. They basically they attempted to treat, this is Martin Klein, they attempted to treat 2 patients with severe beta thalassemia. And there were clinical studies undertaken, but they were actually, what these studies helped, they showed that there was a fear, but that fear should not be there of administering recombinant DNA molecules. But then these people were eventually, they had to lose their job. But again, we learned something from it. And that was good. But then look where we are after. If we had given up there, we would not have come this far. We did AAV, we had AAV in 1965, and then all the way until 2017 when we got Luxturna, approved first gene therapy product. So that was very nice. In between, there were some hiccups again, and that was this 18 dying of experimental gene therapy. Again, the field took a big hit, but then since this is science, we really need to keep going. And that's when, after we understood what went wrong, we again got back up, and now there are so many drugs which are for gene therapy, including drugs in eye diseases, including Luxturna, but there are so many now that are being studied, either studied or they are out there. And we have already heard about clinical applications. There are multiple systems that are being worked on for gene therapy, including ocular diseases and ocular gene therapies. I think this is not new to this audience, but we prefer ocular gene therapy also because it being partly immune privilege, we cannot really say it is immune privilege completely. It needs small volumes, so that helps with manufacturing costs. And many ocular diseases are monogenic, so it becomes easier to look at the primary outcome of the genetic defect. And there is easier accessibility, and by accessibility I mean different routes of administration. This is all very well known. Intravitreal, subretinal, we heard today very nice talks on subretinal injections. We could do suprachoroidal is also being developed now, which could be less invasive. Intravitreal, of course, is going on with AAV and also with small molecules. And this was the success story that happened with Luxturna. No new news for it, but it's just trying to get to that we did have some good things going. And this is the gene therapy landscape. We have now almost, there are just so many diseases, not only the back of the eye, but the front of the eye. We have so many diseases where we can actually get in by gene therapy. But there are some limitations still. We still have to work on. We hear a lot about positives, but we also should be looking into negatives. There are pros and cons for the delivery routes that we take. Subretinal, intravitreal, subretinal is the best way to go for a localized delivery, but then that is localized delivery, and then we have seen there could be challenges. It is a operating room procedure. Intravitreal, suprachoroidal, they have their own goods, and the bads being that there could be possible leakage into the systemic circulation, and that could have an impact on the immune privilege of the eye that we talk about. But still, intravitreal suprachoroidal is preferred, They still need to be tested in the field compared to subretinal injection, which has been very successful in the clinic. And with immune response, I talk about with AAV, it is a virus. If you inject a virus into the body is going to react. You cannot have a time where the body will not react to a virus. So we will get immune response. There is pre-existing immunity and complement-based adaptive immunity. So we have to take care of this. There are drugs which are being given right now, but many drugs actually fail because they are just too much immune response that happens, and AAV is one of them. There was a talk on developing new AAV capsids. Maybe we can develop better capsids which will evade immune responses and still do the work. We also have limitations in animal modeling and how we can translate what we see in the discovery phase, an early development phase to clinic, because a lot of failure happens when we do not really do our due diligence in the preclinical setting or non-clinical setting and then get to the clinic, and that's where we fail. So there are limitations. Animal models are great. We should work on them. We should look at it, but we also need to make sure that when we look at animal models, there is also a discrepancy because we're talking about humans in the last part of this struggle. So there are now bioengineered human disease models which are now coming up. Again, they have their pros and also there are limitations. They are not a three-dimensional organ in a body. And in a typical, as I said, there are just so much that we lose because we cannot really study the disease well enough in patients and then model it in labs in order to make sure that we get the right thing to the patients. The first and foremost being safety and the other being efficacy. Safety is, of course, the first thing we have to make sure. And safety also, there are restrictions. We do safety later. The best model is NHPs, but then it's still an NHP. Immune reactions will differ. So we do best we can. And we still need to keep working on it. I mean, there are again, goods and bads. Hindrances in animal modeling in terms of eye size, that's another big issue that we have to face. But the reason I'm showing these challenges is because it's not only the clinic that has to face these issues of a drug giving a bad reaction in a patient, because that's the worst thing you want to see. But in order to get a drug into the hands of a doctor who is going to inject into a patient, we need to do our due diligence from preclinical perspective that we hand the best stuff over. And it's not going to be straightforward because you cannot work with humans in a lab. So we need to devise better ways and keep devising. There are better ways which are already being done, and we need to keep going. And that's basically where I would like to leave this, that progress is made in both academia and industry. Academia is actually leading the progress in developing new vector, developing new gene therapy perspectives, but we really need to make sure that once we get to the late stage development of a drug, we are doing our due diligence in terms of the big problems that clinic faces. We should all be looking into how can we mimic those problems in the lab so that we can take care of them, whatever we can, we take care of them here before we get it to the clinic so that our patients are safe. I'll stop here, thank you.
Thank you, Dr. Khanna. Any questions from the ground? If not, maybe I could ask one. Sure. Thank you so much for taking us down memory lane, looking at how gene therapy developed. So perhaps, looking back, what do you think was the main technological or scientific hurdle that have to be overcome before gene therapy became something that we could apply to humans? And then maybe looking forward, what do you think are the key technical limitations that we need to overcome in order to make it a more widespread treatment modality?
Yeah, great point. I mean, I think that is what we want to communicate. There were things that were, first of all, the first thing that was really new was I'm injecting a virus. So is it going to be good for a patient, for a human being? You're injecting, knowingly giving a virus to the patient. Of course, you have to educate them that this is not a live virus, this is not what it could do. And that leap had to be taken, that we need to show definitively that like we discussed earlier, was it's not integrating into the genome. It is safer to a large extent, so we need to do those studies to make sure that it is safe. Going forward, I think that's what our clinical colleagues, they are our best source of information. They tell us that we see a lot of inflammation when we inject, let's say intravitreally, or we see a lot of systemic leakage of our virus. How can we make these viruses safer? How can we make these less immunogenic? There is so much work going on already. I think that is where crosstalk with the clinic and the lab is very important to really tell us where the challenges are, so we can start focusing on those challenges. What we hear is, I think, big aspects are inflammation, immune reactions for now, but if there are more, please educate. Yes, thank you.
Thank you. Well, with that, thank you very much for your talk. So, for the last speaker for this session, we'd like to invite Dr. Robert Hai to share with us on gene therapy for ophthalmic diseases.
Good afternoon. Thank you for the invitation to speak today. Some of the excellent preceding talks have made it, I think, a lot of my initial slides here in this talk a bit redundant, but I'm just going to review some of the recent advances in clinical trials for gene therapy in ophthalmic diseases. Just by way of disclosures, I am a consultant for Janssen, which is a sponsor of one of the trials whose published results I will discuss, as my funding is as stated. So we've seen before that there are a number of different ways to approach gene therapy, and gene therapy can be done to either modify or manipulate the expression of a gene or to alter the biological properties of living cells for therapeutics. Depending on the type of genetic disorder and disease, any of a number of strategies could be used to have therapeutic effect, including a gene augmentation or replacement strategy, a gene editing strategy with CRISPR-Cas9 techniques, for example, gene silencing with RNA, or using a gene that can actually modify the expression of other genes, and so it's known as kind of a modifier gene therapy approach. So the following talk is not exactly comprehensive of all these different approaches, but just wanted to highlight some of the advances that may have some of the broadest implications or that are farthest along currently. So we've already reviewed some of the history of gene therapy by the preceding excellent talk, and it's amazing to see where we are after maybe 30 years of research, from first being able to identify viral vectors for gene delivery to the development of a therapeutic with Luxturna, which, as we know, has been used to treat LCA due to biallelic mutations in 2017. And so there's been well over 1,000 active clinical trials using gene therapy, and we have potential treatments for the previously untreatable. Luxturna, as mentioned, was approved in 2017 and is the concept here is subretinal gene delivery of RPE65 using an adeno-associated virus. And it's really been a benchmark for how future trials have been developed, primarily because of its success. It's shown durable improvement on, for several years after the initial trials. The outcome in the initial trial was a multi-luminance mobility test, which was something that had to be really developed for a specific function that was being assayed here, which was improvement in rod photoreceptor function. And remarkably, many of these patients showed improvements immediately after treatment, and those were sustained after 4 years. And other improvements were also noted in perimetry and full field light sensitivity thresholds. So the majority of the trials in ophthalmology have focused really on a gene augmentation strategy. And as you can see here, there's been, of all the trials, many of them have been focused on that strategy, but others have also been used for other types of diseases. including not only retinitis pigmentosa and Leber congenital amaurosis, which are primarily diseases that affect rod and RPE cell function, but other types of diseases included Leber hereditary optic neuropathy, choroideremia, X-linked retinoschisis, and so on. But to date, we still only have one FDA-approved gene therapy for an inherited retinal disease. Gene therapy for X-linked retinitis pigmentosa due to variants in RPGR, probably among all inherited retinal diseases, has shown probably the most significant and meaningful effects in several large clinical trials. RPGR accounts for 70% of X-linked retinitis pigmentosa, or about 1 in 40,000 males. It usually is more severe than other forms of RP, with children affected usually with the first decade of life. And phase 1-2 studies sponsored by Nightstar and Mira GTX for subretinal delivery of RPGR were promising, and these companies were then acquired by Biogen and Janssen, respectively. Beacon Therapeutics has another trial, and 4D Molecular Therapeutics had one that was recently discontinued. So I just wanted to briefly highlight the results of the gene therapy trial that was conducted by Nightstar, now Biogen. This was a phase 2-3 study of patients aged 10 years of age or greater who were treated with subretinal gene delivery using the AAV8 vector. There were about 29 total participants. And the primary outcome was microperimetry median responder criteria, which meant more than a 7 decibel gain at 5 of 16 central loci. You can see the kind of represented images of the microperimetry data in the untreated and treated eye. And you can see that that was quite a significant change, but unfortunately the study did not meet its primary outcome. Secondary outcomes were also encouraging, though, and some patients showed durable gains in retinal sensitivity far after treatment, so somewhat reminiscent of the Luxturna experience. Janssen is now conducting a phase 3 study of subretinal delivery of RPGR using AAV5 in 97 patients with X-linked RP. The primary outcome is change in performance on a vision-guided mobility assessment akin to the maze used in the Luxturna trials. There are many secondary outcomes, including retinal sensitivity and questionnaires, as is the usual case, but there's reason to be optimistic about both sets of outcomes based on their earlier trial results. And in the earlier phase 1-2 trial, there were significant improvements in the primary outcome, which is a maze, at 26 weeks following treatment. And as you can see in the chart on the left, treated patients took far less time on average, about 37 seconds. Sorry, the pointer's not cooperating. In the maze that mimicked deep twilight, so the darkest conditions that these patients were asked to perform. And there were also significant improvements in secondary outcomes, including static perimetry and microperimetry that were reported, and it's tabulated on the right. Beacon Therapeutics is currently conducting its phase 2-3 clinical trial. The results are obviously not available. Some of the earlier results were announced at the annual meeting in Chicago in 2024. Here it's, again, subretinal delivery using AAV2, and the response was a microperimetry change greater than 7 decibels in at least 5 loci. And at 24 months, they showed that 4 of 7 study eyes showed a response in the high-dose arm, and fortunately, no clinically significant safety events and treatment emergent adverse effects were mild or moderate. Switching to the gene modifier approach, this has been something that's been pursued by a company called Ocugen for all forms of retinitis pigmentosa. So in their clinical trial, they're testing the hypothesis that all forms of RP may be potentially treatable with a subretinal delivery of the gene NR2E3. Currently, they have a phase 3 trial underway, which is associated with, trial underway in which they'll have half of the patients who have a Rho mutation and half the patients who have a gene agnostic mutation. And the idea here is that perhaps by modifying the expression of other genes, including Rho, CEP290, and so on, they might be able to promote a retinal homeostasis that can improve retinal function and retinal sensitivity. The primary outcome, much like the other trials, is a gain in lux levels on a luminance-dependent navigation assessment akin to a maze. And it was their earlier trial that was a phase 1/2 that demonstrated about 78% of eyes improved on that test. And among patients with row variants, 80% demonstrated preservation or improvement of vision. Optogenetics is a newer player in the field when it comes to a mechanism of action for visual restoration. There are several sponsors that are currently investigating the potential, including GenSight, Bionic Sight, and Nanoscope. Here the idea is you have a light-activated photo switch that targets non-photoreceptor cell types in the retina. And again, it's a gene-agnostic approach that can potentially improve vision in patients regardless of the underlying genotype. Nanoscope reported last year the results of its phase 2 trial using a multi-wavelength cone opsin delivered intravitreally in patients with RP, and there were significant gains in vision at 52 weeks with a mean increase of 0.34 logmar compared to sham, and at 76 weeks, both gains in vision and the proportion of patients with more than 0.3 logmar were significantly elevated compared to sham. There are currently 4 different gene therapy approaches underway in phase 1/2 trials for ABCA4 retinopathy or Stargardt, and a handful of other phase 1/2 trials due to RS1, PDE6B, and MYO7A, although the results for many of these are obviously forthcoming. Among them, however, there was a remarkable result recently with the gene AIPL1. AIPL1 associated dystrophy is associated with very limited light perception at a very early age, and the idea that you could potentially treat patients within a very narrow window was supported by their findings that in 4 children that were treated, vision improved dramatically from 0.8 to 0.1 logmar from essentially light perception. Other outcome measures showed strong treatment effects, including improvements on VEP, functional vision, and retinal structure, although many of these assays were specifically designed or modified for measuring vision in very young individuals. So thank you for your attention, and just to conclude, there's a lot to look forward to in gene therapy for ophthalmic diseases.
Thank you, Dr. Hyde. Any questions from the crowd? Please.
How can we enroll our Indian patients into these trials, if it's possible?
Well, that is a great question. I think it is a matter of discussing with the sponsor and getting early involvement with sponsors and indicating that you have an infrastructure available to support clinical trials. The idea of conducting international or multinational clinical trials is one that I think many academic ophthalmology departments are pursuing in conjunction with industry. But I think it's one that I'm, this is the reason I'm here in India, to consider how we can better establish relationships to make these types of things happen.
Thank you.
Yep.
Hi, nice talk, and particularly, although the results are mixed, but obviously it gets refined over a period of time. I was wondering about the last gene, the AIPL1, and with one of the patients with 3 different variations, like you had a missense, you had a duplication, as well as a frame shift. So what was the actual structure of the gene? Because when all of these are occurring together, what's the final end product which is being targeted? Because the missense is there, but prior to that there is a duplication, and then a frame shift, which obviously would lead to a termination at some point of time of the protein. So what's the protein that is being targeted, and what was the outcome?
Yeah, so I think that's a great question. Obviously, I think you're speaking to the idea that there could be nonsense-mediated decay from early variants in the sequence. And to answer your question, frankly, I'm not sure. I can't speak to that very well.
I just had a question about, you mentioned NR2E3, to sort of use that gene or express it to treat any other forms of gene-associated disease, right? So is it? I presume then the action is that it generally stimulates the transcription of a number of photoreceptor genes. Is that how it works?
This is a very, I don't want to say controversial topic, but I think it is one that is very complex. NR2E3 and NRL are both transcription factors that modulate the expression of many downstream genes. That work by Nina Hayter in preclinical models used a number of different gene models, including CEP290 null, Rho null, and so forth, and showed that in response to delivery of NR2E3, there were improvements in ERGs, as well as on a whole host of transcription factors that are thought to be important for maintaining retinal function. It's a bit of a hand-waving explanation for what is probably a very difficult or very complex question involving a network of neural regulation.
Yeah. Any further questions? Okay, I think if there are no further questions, we will end here. Thank you very much. And I would like to thank all the speakers for providing various, I think, right from historical, the present state of the art and future perspectives on this very challenging field in which we obviously still have a long way to go, despite the RP65 story. Thank you.